Hi ISMB, my name is Luca, and I am going to present my poster, Ploidetect enables pan-cancer analysis of the causes and impacts of chromosomal instability. So basically, cancers accumulate copy number variants, and these are just changes in the quantity of DNA at different regions of the genome, and this happens because of a process called chromosomal instability. Chromosomal instability and copy number variants are fairly important in oncogenesis, and there's a lot of different biological processes that interact with chromosomal instability. Right now, in a lot of cancer research, people are sequencing a lot of tumors, and because this is becoming very popular, it's really important for us to be able to identify copy number variants in tumor genomes with a high degree of accuracy in order to get the most out of these large data sets. So with that in mind, what we aim to do was to develop a better copy number caller than what already exists, and apply it to learn new things about chromosomal instability from the copy number data, and also just to see which copy number variants we can find that occur often in cancer. The way that we did this is we developed a tool called Ploidetect, and basically it uses some statistics to first determine tumor purity, which is a common um, nuisance factor in copy number analysis, and then we perform segmentation of the copy number data using a novel segmentation algorithm. Then we take these segments, we assign zygosity, we assign copy number, and we get a copy number profile. We applied this to 725 sequenced pretreated metastatic tumors in the personalized oncogenomics program, and we compared the results to three other tools, Sequenza, Purple, and BigSeq2. So first of all, we looked at um, the tumor purity estimates of Ploidetect, and we compared this with some manual review data. So basically, we had expert bioinformaticians sit down and look at copy number profiles estimated at different levels of tumor purity using a copy number color. And they essentially decide on what sounds like a good estimate of tumor purity. And what we found is that the estimates that Ploidetect gave us for tumor purity agreed really well with these manual review estimates. And when we went back and, with the Ploidetect results in mind, manually re-reviewed everything to look for alternate models, the results really didn't change all that much. Then we also decided to look at an in silico dilution of the colloid 29 cell line data, and we found that there was very good agreement between Ploidetect's estimates and those. Furthermore, we then took a look at comparing the actual copy number results between Ploidetect and all the tools in our large cohort, and basically we found that Ploidetect was able to detect more cancer-related copy number variants than any other tool, and also the segments were on average larger than all of the other tools segments. So this indicated that Ploidetect was able uh, to generate fewer false positive segment breaks while also maintaining the ability to detect small, focal, or hard to detect copy number changes. We manually reviewed a selection of the data for each tool, and we found that Ploidetect had the best precision for homozygous deletions. It didn't make a single mistake in the 100 copy number variants we looked at, and it had the second best precision, but much better recall than the next best tool. Then we decided to look at the actual results and what we could learn from this. So we first looked at chromosomal instability using the segment N50 as a proxy. We found that there was a association between chromosomal instability and the tumor mutational burden. We found that different tumor tissue of origin led to different degrees of chromosomal instability. We found that tumors that were more chromosomally unstable tended to be treated for longer durations on platinum, so they tended to have a better response. And we found that chromosomally unstable tumors had reduced CD8 T cell scores and reduced T cell infiltration scores. Finally, we looked at the association of different genes' mutation status with chromosomal instability, and we found two genes that were fairly interesting. There was ROCK2 that impacted mutations of ROCK2 caused chromosomal instability in a manner that seemed to be acting in the same pathway as TP53. However, when we looked at a long non-coding RNA AC074391.1, mutations in this long non-coding RNA actually caused additional increases in chromosomal instability beyond what TP53 alone caused. Finally, we looked at recurrent deletions in metastatic cancer. So we did this by randomly shuffling the positions of our observed homozygous deletions in our cohort, and basically observing which positions were deleted in our cohort more often than by random chance. And so we found a lot of the usual suspects, a lot of commonly homozygously deleted tumor suppressors, but we also found a number of fragile fragile site genes that only lost specific exons, and many of these are still known to be cancer related. There was one gene that we chose for further analysis, but there wasn't really room in the poster to expand on this anymore, but you can ask me about macro-D2. What we learned, essentially, this is just summarizing what I've already told you, so you can read it at your own leisure. But essentially, the point that we're trying to get across here is that what we've done is show you that if we can do a better job of calling copy number in cancer genomes, we can find a lot of really interesting things. And unfortunately, the state of the copy number field right now is that copy number callers are not nearly as accurate as small variant callers. And if we can try and narrow that gap, there's a lot that we might not understand yet about how copy number behaves in cancer genomes. And we hope that this is a good demonstration of what we can do with better callers. And so here's our acknowledgements. Obviously, we acknowledge POG program, the POG patients, their families, and we never would have been able to do this without them. And there's a list of all the funding sources for POG and also for myself. And here is our affiliations. So thank you.